So let's talk about the tree of life, the tree that grows from maternal blood and creates a baby. Its leaves are taking oxygen and nutrients from the mother and are driving them to its roots, where a baby is growing. And that is the placenta. I'm not sure how many of you have ever realized how important is the placenta in your life. Each and one of us comes from a unique and personal placenta. And if you look around, you'll see how different we are. So you would expect our placentas to be as different as we are, because each placenta is growing a completely different person. And so the health of the placenta not only dictates the health of the baby in utero, but also the health of all of us as adults. There are some pathologies of the placenta which have been directly related to cardiovascular diseases in adulthood and even to diabetes. The placenta is an organ that undergoes massive changes under different physiological conditions. In just 9 months, it develops, functions, and dies. During the first trimester of pregnancy, the placenta is developing together with the embryo in a low oxygen environment. There are plaques blocking blood flow from the uterus to the placenta, and the embryo is feeding from the yolk sac. Starting from around week 13, blood starts to flow into the placenta and it starts to function. The placenta continues to grow to meet fetal demands until delivery, when it's delivered together with the baby and thrown away as medical waste. Yes, you heard right, it's thrown away as medical waste. The organ that gives us life is thrown away in a matter of seconds. Now, if the placenta is such a crucial organ, then why is it the least understood of all? Well, in vivo research into the placenta is mostly constrained by ethical and safety issues, not to mention the fact that pregnant women are not willing to take part on any research. Now, ex vivo research of the placenta is tricky. The placenta is a highly vascular organ, is mostly composed by blood and blood vessels, and so quickly after delivery it collapses and loses its morphological information. But not only that, we have access to the placenta only at delivery. And so everything that happened during 9 months stays a mystery. Now, animal models are difficult to extrapolate because each placenta is growing a completely different animal. And so they not only differ in their size and their anatomy, but they also differ in their biological and biochemical properties. And so really, there is an urgent need to develop new technologies to investigate the human placenta. Now, you see, I'm a mechanical engineer by education. And so I really think about the world in these terms. So that is, how can we make this machine as efficient as possible? What is the minimum energy that I need to invest in order to obtain my goals? Or what are the forces controlling the movement of these machines? And so when I was first introduced to the placenta about 10 years ago, I was amazed by the fact that a placenta is actually a biological machine. It works very similar to a heat exchanger, where heat is transferred between two fluids flowing in different directions. And so then I realized that mechanical forces must have a role in the regulation of the transport of substances between the mother and her baby and the baby and mother, but also in the development and health of the placenta. But not only that, probably mechanical forces have an impact on placental pathologies and pregnancy complications. And so, really, that is the field that I'm interested in. At the macro scale, it's called biomechanics. It is the study of the structure and function of a biological system using concepts from mechanics, which deals with the effect that forces have on the movement of both tissue and blood flow. In this example, I took microscopic images of the fetal placental vasculature and created models of blood flow and oxygen transport, revealing that the complex and unique architecture of these vessels are actually promoting oxygen uptake by slowing blood flow exactly at the locations where the membrane separating the two blood circulation is thinnest, so that oxygen crosses quickly and then has enough time to bind to hemoglobin. At the macro scale, this field is called mechanobiology. Mechanobiology deals with the role that physical forces and mechanical properties of cells and tissue, that is, for example, elasticity, have on the development, cell differentiation, and disease. Now, perhaps the best example of mechanobiology is the Nobel Prize awarded in 2021, which was awarded for the discovery of the mechanoreceptors in our skin, those receptors that can feel mechanical forces, such as pain and touch, and convert them into a biological signaling pathway. Now, there are many experimental techniques to investigate biomechanics and mechanobiology. But in the placenta, surely they are non-existent. And so, what if we can create a virtual placenta? What if we can create a platform where we can perform experiments that are otherwise impossible in vitro or in vivo? Let me explain you what I mean. Let's say I have a cake with 20 ingredients, and I want to know how each of my ingredients are affecting the outcome of my cake. So one approach would be to bake 20 cakes, leaving out one ingredient at a time. But this is time-consuming and probably money-consuming too. But what if I can make a cake in my computer, exactly the same cake, and I can mimic the baking of the cake? And so in a matter of seconds, I'm going to have all the 20 results. Now, in the computer, I can even do the opposite question. I can tell my computer, I want to have a softer cake. Please give me all the possible combinations and by how much I need to adjust each of my ingredients to get a softer cake. And I'm going to get it in a matter of seconds, too. And so that is what I do, what I've been doing for the past 10 years. I create computational models of the placenta to investigate, analyze, visualize, quantify the role that mechanical forces have on placental function. In this example, we created a model of a placental fetal exchange unit, and what we found is that what is driving blood into the placenta is not the strength of the blood, incoming blood. It is actually the density of the tissue. Well, not the density of the tissue, the changes in the density of the tissue, which are creating local pressure gradients and are driving the blood deeper into the tissue. And so, just to conclude, computational biomechanics is an emerging field that can help to elucidate on the role that mechanical and physical forces have on placental function, in my case, and of course to help and elucidate the pregnancy complications. And so, last but not least, I would like to thank everyone who has ever been involved in these projects, to my amazing supervisor, Professor Michal Neiman, and please don't eat your placenta. Thank you.
Thank you very much, Romina, for this fascinating fascinating talk. We have questions. Yes, please.
Thank you for a fascinating, really, lecture and something to learn about. I would like to ask where the ethical, are the ethical problems or the notion of surrogacy, pundekaut. How does it come into it? Pundekaut.
Pundekaut?
Yes. Because then you have the placenta of another person, and how does it influence all the procedure?
Interesting. So first of all, the placenta is a fetal organ. So, I mean, even if you get from pundekaut, you will get the placenta of the baby. So it should match, at least in most of its part. But also, I have to say that I never got a placenta from pundekaut. So I've never had the opportunity to think about this problem or this scenario.
I just think about the influence of the placenta. How does it influence, the whole, development of the baby?
Well, I don't think it will be any different from any other pregnancy.
Mm-hmm.
I think it's the same role that a maternal uterus has. So as long as it's healthy, and hopefully, it will be a healthy placenta and a healthy baby.
Thank you.
The thing with these models is that you have really 3 blocks to make them realistic. So you have the geometry that needs to match as much as possible. Then you have the mathematical description, which are the equations that you are solving. And then you have all the external forces or environment that is being applied on your system. Now, I think that the most consuming part is actually the geometrical description, because the placenta is highly variable, even within each placenta and between placentas, as we are. And so really there's still no average placenta. We still don't know what is healthy as an average. So the most difficult thing is actually to generate the geometry of these models. Then to run the simulation in one day, or a very complex one. In terms of imaging, probably machine learning is going to get a role here. But I also assume that with time, then solving these problems will not be numerically specific per se. Let's solve each equation, but it will be estimated by machine learning. I think that's where the world is going, so probably.
From conclusions from your experiments that you are performing, can you manipulate the placenta if you know where is like the problems or what is like covering something?
No.
Can you know? It's not like yet.
We cannot even know where the problem is yet. So.
After your research, we will know more.
Hopefully.
So let's say thanks, Romina.
Thank you.